Well, thank you so much for being here. It's really a pleasure to come tell you a little bit about my work, the bacteria that I care about, what I'm really interested about, and what my group of fabulous students are doing. Many of you probably met them outside. They had a lot of the microscopes, and they were showing lots of plates, and they were really excited to do that. So, muy buenas noches a todos y a todos los que están viendo el programa por su computadora también. If I had to take a guess of what your first encounter with microbes has been, or maybe even what your current relationship with microbes is, I think I will bet that this video probably summarizes it pretty well. Okay. There we go.
In a remote African jungle, a small monkey is captured. Bound for a pet store in America, the animal carries a deadly virus.
Now, I know that some of us have doubts about what we're about to do. We'd be less than human if we didn't. But the fate of the nation, perhaps the world, is in our hands. We cannot, we dare not refuse this burden. I'm confident that each of you will do his duty. God forgive us.
Your town is being quarantined.
We got 19 dead. We got 100 more infected. It's spreading like a brush fire.
What are you talking about?
If one of them's got it, then 10 of them have got it now. And if one of them gets out of Cedar Creek, we have a very interesting problem.
If that bug gets out of there, 260 million Americans will be dead or dying.
I'm leaving with the team in an hour.
From the heart of a small California town.
Damn it, Sam, I want to save these people same as you.
To the inner circle of power in Washington.
The most optimistic projection for the spread of the virus is this: 24 hours, 36 hours, 48 hours.
The greatest medical crisis of all time.
We can't stop it.
Begins.
Try to remain calm.
Many people are dying and are going to continue to die unless we find this monkey.
There will be panic the likes of which we have never seen.
There you are!
Attention, Portland residents, citizens of San Diego, residents of Atlanta, Denver residents of Detroit, attention, Dallas residents of Chicago.
So was I right? Okay, that was a pretty good guess. And the reason why I took that guess is because when we think about microbes, they're words that we immediately think about. Fear, anxiety, terror, risk, there's a panic. Something has to happen quickly, otherwise humanity disappears. And actually, it turns out that a lot of our American voters feel the same way. In fact, a couple of years ago, when they were polled, 58% of them said in 2009 that one of the things they worry a great deal about when they think about microbes, the first thing they think about is contaminated food. Right? A couple of years later, in 2013, actually the percentage grew of people that said that their first impression of microbes, it's that they worry a great deal about infections and antibiotics. And so I just want to tell you that I have been very fortunate since I was in middle school, like many of you in the audience, from a very young age to encounter teachers that really cared to show me a different side of the story. So through going through middle schools and science fairs, I sort of became really interested in these tiny little organisms because they can actually carry life. And that idea and that sort of curiosity about how they do it, being so tiny, it's really what has fueled my interest in microbes for a long time now. And so what I want to talk to you a little bit about today is sort of our discovery of microbes, the diversity of microbes. And then I want you to walk away with one of my favorite microbial stories, which is a special guy that I will talk to you about today, one of my favorite bacteria. And so when we talk about the discovery of microbes, one of the things that we should keep in mind is that even if we go 4,000 years back and we think about the Mayans who lived in what's now the area of North Mexico and Honduras here, one of the things that we knew is that they really knew how to feed their people. right? They never really ran out of food. And one of the reasons for this is that they had very sophisticated agricultural systems. In fact, one of the things that they invented for us that we still use to this date is this notion of crop rotations. And what they mean by that, or what they meant by that, is that it's pretty much what we would say, as my husband will tell anyone interested in planting plants in our garden, is not to plant the same crop in the same soil location every year, right? And the reason that this happens is, or what the Mayans figured out a long time ago, is that if you take a crop 1, crop 2, and crop 3, and you plant it in a very specific location of your soil, the next year you probably wanted to rotate it. So you wouldn't plant crop 1 again, but you would do 2 and maybe 3 and maybe 1, right? Now, back then it wasn't very clear why, but there was an interesting clue that this ancient people got. And it's the fact that this did not happen, it wasn't a benefit to the soil just for any random plant. There were actually very specific plants like peas and legumes and lentils that really allow the soil to be a lot more fertile. And so years later, we wonder, well, what is it about these plants that can actually lead to richer soil? And it wasn't until 1683-ish when the microscope was invented by this Anthony Leeuwenhoek guy, who many of you might have heard of in school. And just to give you some kind of perspective, this is at the same time frame where Newton was working on gravitational laws, right? And so this framework was being set there of people were wondering about big things in the universe, but also tiny little things. And one of the things that we can do with the microscope is now see things that we couldn't see before, 300 times smaller than we couldn't see before. So using this novel technique at the time, believe it or not, I mean, now we see a microscope and it's like no big deal. But back then, what you can do is you can really examine and look at these plants and try to figure out, well, what's so exciting about them that they're leading to richer soil. And one of the things that people notice is that they have these little pockets on the roots called nodules. So then the next logical question was, when we started really thinking about this, was, well, what is in these little pockets? And what you can figure out is that in this little pocket, what we are able to visualize are actually bacteria. So what these special plants have that lead to richer soil, it's bacteria in these little pockets. And then you go one step farther and you ask, well, what are the bacteria doing that are actually helping to create more fertile soil? And it turns out that it wasn't even 200 years after that, in roughly about 1889, where another scientist started to culture these bacteria, bring them to the lab, and figure out what they were doing. And what they figured out is that actually they were converting nitrogen from the air and making ammonia. And one of the things you will probably ring a bell to you is that ammonia is what we use today as fertilizers. So the end of the story here is that since a long time ago we've known that there's something special about microbes. In this case, it's not anything having to do with the plants themselves. It's actually the bacteria in the roots that do something special in the soil here to produce nutrients and fertilizers that will help to make our plants grow a lot better. And so, of course, this is something very useful for us today. And then the other thing that I wanted to share with you when we still think about even 8,000 years back, to a completely different part of the world in Africa, the Egyptians knew a little bit about bacteria as well. So one of the things that they were very glad, and probably you're also equally glad that they figured out, is this whole notion that when they dump in a piece of bread, moldy bread specifically, in some water, and they tried, risky enough to try that mixture, they call this the joy bringer drink, right? And maybe a lot of you guys will still think about it as a joy bringer, but essentially what they were doing is we know back from the writings of even that long ago that they were making wine and beer. And of course, as you now know, and as we now know, what's going on here is that this bread is moldy, and the mold, the yeast in this bread, is actually converting the sugar in the bread and making alcohol by a process called fermentation. Okay? And still today, this is something we use to make lots of flavors of drinks that a lot of people like, some more than others, right? And you can combine grains and malts and hops and fruits and make a whole variety of beer that we use today with different bacteria, yeast microbes in this case. Okay, so what we learn here from looking back at even these ancient cultures is that without knowing it, these cultures were not lovers of microbes. they were not fans of microbes. Without knowing it, they really became dependent on microbes to feed, entertain, have fun, wine, and dine their populations. So what are these bacterial microbes? So far in the talk, we've actually covered, maybe you noticed this, maybe you didn't, the main microbes that are talked about, 3 completely different classes of microbes. So in the video, we talk a little bit about viruses. In my example of these trees here and the soil, we talked about bacteria. And in this latest example, we talked about yeast. It turns out again that these are very different microbes. It's like a completely new world of living that these guys represent, which is why a lot of people tell you do not take antibiotics to kill the flu, because the flu is a virus, and antibiotics will kill bacteria. Completely different things. But one of the things that we know is that they also act very different. So for example, viruses, the smallest of these 3, they really need their host to sort of survive. They need to infect something to survive. So they're not completely independent on their own. Bacteria, which is really why I'm truly fascinated by them, are one of these tiny little forms that now are completely independent. And then yeast are a little bit larger than bacteria, and they're also independent and can do some more complicated things than bacteria. And so today what I want to tell you, it's really focused on bacterial microbes. So before we go any farther, what I want to really get across to you is how tiny these organisms are. So what I want you to do is take a piece of your hair, and if you don't have long hair, find a quick friend around you with long hair, and then turn your piece of hair around you and look at the tip of your hair. Just look at it straight up at the tip of your hair, okay? And you probably are not going to believe me when I tell you that 100 bacteria can fit on the tip of your hair. Now, these are 100 jelly beans. There's a lot of them. So that's how tiny these things are. Again, tip of your hair, turn it around, you can barely see it. This is how many bacteria we can fit in there. So I'm going to pass these around. I guess the last row can eat the jelly beans, but while we get here, we might not want to do that. And so the point is they're very tiny. So one of the things then that all of us bacterial fans worry about is what is it that these organisms do to stay alive? And one of the nice things about them is that they actually do a lot of the same things that human cells do to stay alive. They have a membrane, so they have a little skin that protects them. They have a little flagella or pili where they actually are motile and can move around. They eat a lot of the nutrients. In fact, they are less selective than we are about our eating and our diets. They eat a lot of different stuff, and they reproduce very fast, right? And so one of the other interesting things here is that then when we think about, well, how do they do all these things? Again, like humans, a lot of what they do is encoded or programmed, or it's based on information that is contained in their DNA. And so when we look at DNA, and we are very familiar with this image when we think about our own DNA, what we're really looking at when we say the word genes are these little pieces of this DNA. So every little piece here is what we call a gene. And DNA, as of course we kind of figure, contains a lot of information. So what I want you to remember is that something that is not as intuitive about DNA is that this information is there, but it's not useful until you actually process it. And there's lots of processes to do that. 2 of them are transcription and what we call translation. But the point is that through these processes, they actually become active. So it's not just enough to, they actually have to be activated. And when they are activated, that's when life gets fun, and we get diversity, and we get brown eyes and black eyes and thinness and muscular and smart and anything, all the traits that you can think of. And in the bacterial world, I guess you're happy if you get thicker membranes or thinner membranes, or you get longer tails or shorter tails, right? But these are traits. So our DNA encodes all this information. It has to be activated, and then we can actually use it in a functional form of traits, okay? And this is what's kind of summarized here. So it is this scheme here, the information that we contain and how it's actually processed into something active that leads to a lot of the diversity we see. Now, there's another good news that you would appreciate, and it's the fact that a lot of these bacteria, they do the same processes to activate their genes and to encode their informations than human cells do, which is why we can kind of convince people, one of the reasons that it's not just that we like to study these things to make them do stuff like wine and beer, and so that we can feel happy about ourselves, right? It's also that we actually learn a lot of basic biology functions that apply to human cells. So a lot of what we learn about bacteria and microbes actually applies to the trillions of human cells that we carry as we move around. So the next thing that you probably are wondering is, well, how many bacteria are there? Right? How many families are there? What is this world like? And one of the things that will probably surprise you is that actually, I don't know. We don't know. There is estimated that there's about 2 billion families of bacteria out there. Those are 2 billion different bacteria that can do 2 billion different things potentially, right? And so far, we've met 1% or less is the estimate. That's if you estimated that there were all these people in the world. And of course, as a New Yorker, I'll say, and all you know, it's people that live in New York. And Jay is giving me the good thumbs up here. Okay, but that's the same amount of knowledge we have about bacteria. As if all we knew were New Yorkers, that one little section of the world, and we knew nothing else. 1% of the bacteria is at most what we know. The other interesting thing is that we might not ever get to know them all. Because for us to cross to the bacterial world, we have to bring them to our labs. It's a lot harder to go and find them in their natural environments. And we may never be able to bring them back and actually recreate the environments where they will grow happily and replicate. So we may never actually be able to study all of these. And so the point is that when we have all this information and how we process it, not only do we create all these diversity, but actually the variety that is encoded in the DNA and how it's activated leads to all these different traits that are the traits that we exploit when we think about what to make useful with these microbes. Okay, so you saw there was a nice table out in the fair earlier, and you saw lots of products that are associated with bacteria. Some of them are vinegar and insulin. We use bacteria to make. Cheese, there's about 1,000 types of cheese that I think we now have, and each of them use different processing conditions and different organisms. Buttermilk, biofuels is a hot topic that you might hear in the news to create cleaner energy sources. And something that is also interesting is plastics. We actually make a lot of plastics using bacteria because they adhere to their surfaces, and that same plastic we can use for something useful that we like or that we want to make cheaper. And to clean our environment. A lot of these bacteria, as I said earlier, they're not very selective on their diet. So you give them metal and they'll eat it. And not only will they eat it, but they'll make something useful with it. So they're the perfect candidates to go clean the environment for free and give us something to make money out of, right? So that's why they're big fans of, we're big fans of them in that way. But then still, right, we struggle, and there's still this huge negative perception of microbes, right? And we saw, right, people out to kill them, right? That's what we think when we think about them in the big screen. And, you know, to set that record straight, this is not totally unjustified, right? I mean, they are terrible microbes out there. They tend to have this habit of making people sick. And so some of the ones that I would completely advise that you stay away from would be things like superbugs, which are completely resistant to any antibiotic that we have. Things like gangrene, tuberculosis, gastroenteritis, all of these are bacteria, and the list is a little longer than that I would personally stay away from, right? One of the ones that you might recognize is the Black Plague. And this is for those of you that are Shakespeare lovers. In the times that Shakespeare was writing Romeo and Juliet, this bacteria right here is responsible for wiping off 1/3 of Europe's population. Right? It spread quickly in trade routes, and it actually killed 1/3 of Europe's population and a lot of the rest of the world. And it greatly affected a lot of our favorite writers. And if you read carefully, even Romeo and Juliet, part of the reason that letter was never delivered is because the priest had to be quarantined because this disease was going on. Right? And so we didn't really know it at the time, but now we know that this is caused by a bacteria, Yersinia pestis, who can actually infect the human stomach and paralyze the immune system. And so the reality is that microbes are actually pretty happy coexisting in the human body, and we're actually happier that they're there. So it turns out if you take our bodies and you do a tally of what the types of cells that we have in our body look like, if you condense all the human cells, this is about the fraction that we have, right? Doesn't even make our entire head. So if you take all of the human cells and you put them all together, there is a tiny little fraction of us that's actually human cells. The rest of them are microbes. They are bacteria that actually keep us going. A lot of them are in the gut, a lot of them are in the skin, and you'll be happy to know that these are perfectly beneficial bacteria, that most of them are kept in check by our own immune system. It's a really nice collaboration that they have with human cells to keep us going. And in fact, when the balance is off, a lot of us start getting sick, and that's how a lot of the sickness starts to arise. So bacteria, it's all over us, many, many different kinds all over our bodies. We don't exactly know yet all the benefits of every single class and every single type and what they do, but we know at least two very powerful examples. One of them, it's the bacteria that we have in our intestine, who's responsible for about 75% of the vitamin K that we get, very important cofactor for keeping our blood the way it should be kept. And in our gut, there's also a lot of bacteria that help us digest a lot of the plants we eat, like vegetables. Okay, and so you'll be looking forward to this. You'll see it more and more coming. There's a lot of interest in this research in terms of the ecosystem that we're carrying as we're moving along every single step of our lives. There's all this inner ecosystem that we're still trying to figure out what it means, what it does, when does it get off balance, and what does it mean to disease and to health. Okay? And so the reality then is that there's really a small number of bacteria that get all the press. Right? So the bad players really get all the press, and they're very tiny percentage. Right? In fact, even if we take the amount of microbes, bacteria that we know, less than 5% of them actually cause disease. Okay? So there's still, however, a lot of effort to try to kill them. They are chemicals. Right? We buy anything that says antibacterial, we'll sell. Right? We are going to buy it, we're going to invest, we're going to wash our hands with it, we're going to protect ourselves, and of course, right, then all these physical ways that we attempt to keep bacteria from growing and keep them pretty much or kill them. Right? That's why we ice our food, or we put a lot of salt on the fish that we bring back from the lake when we go fishing, or we eat dry foods, or we will boil water to keep microbes out of it, or we will refrigerate it, or we would do all these things to pretty much slow down the growth of bacteria. Okay? Now, one of the things that I also want to tell you today is that there is some good and bad news with this, right? The good news is that those strategies are great for the most part. The good and bad news is that there's a lot of organisms that actually you won't even touch by any of these conditions. They can survive a lot more things than any human cell can survive. They can survive a lot more things than anything that we know in the world can survive. Okay? And this is not a completely foreign concept. Again, we hear a lot about these in the news when they kind of come and freak us out about bacteria spreading quickly in the hospital and antibiotics can kill them. So these are bacteria that cannot, right, that survive conditions that we wouldn't expect them to survive. Right? And especially what I want to tell you today is about a group of bacteria that we call extremophiles. So these are bacteria that can survive very, very extreme environments. So what do these extreme environments look like? It could be extreme cold. So these are bacteria that if you put food in the refrigerator and your bacteria is already sitting there, you won't kill it, you won't slow its growth if it goes as to many temperatures, as cold as you can probably imagine. There are other bacteria that can survive boiling water. So you can boil your water all you want, but if you have one of these extremophiles that can actually survive such high temperatures water, there's no point. You will burn off your water, you will boil it all off, and you won't kill them, you won't slow down their growth. Right? Extreme deserts, this is actually an interesting one. If you put them out there in the dry, I mean, we are in Texas and we're out there for an hour and we're ready to dehydrate, right? These guys live out there in the desert, and again, they can carry out life. They love it. This is their home. This is what they know how to do. And the last one that I'll tell you is this extreme acidic condition, which again, these are things that we kind of know because we use lemon and we use vinegar to clean our meat, and we think that, you know, yes, this should kill all bacteria. But in fact, there are bacteria that their specialty is to survive these very low pH situations, very acidic situations. Okay, so these are extremophiles. They're out there, and so what's the big deal, right? You might be wondering. And what I want to convince you of today is that part of our interest in this diversity of microbes is because of these type of guys. It's guys that can really do the unimaginable, so that we can really understand, right? Sort of the limit of life as we know it, right? How are these guys surviving the situations? So this is why we call them sort of nature's ultimate survivor. You want to go to space, let's ask a bacteria what it does well so that we can use some of what it knows how to do to carry us forward. And they thrive under these conditions, and we still have not figured out the secrets even for our own living. There's a lot that we can learn from them. So I want to introduce today the special guy I want to tell you about, who's one of the classic examples of these extremophiles. And this is a guy that our lab studies quite a bit. His official name is Deinococcus radiodurans. I recognize that I will stumble every time if I have to say that. So we're going to call him D-Rad for the night. And I want to tell you a little bit about what he does and how understanding a little bit more about this particular bacteria can really have major implications to our own human life. Okay? So what does this guy do? His story starts about 60 years ago in Oregon. Okay? And this is pretty much when there was a random meat facility that was trying to sterilize meat so that they will sell it and so that the meat wouldn't be toxic or wouldn't be harmful or wouldn't cause disease to anybody that would buy it. So this meat sterilization process, any food sterilization process, is one of the most robust things we have to pretty much kill all bacteria. Right? And little did we know it, when these people look at the meat again, what they notice is that the meat was getting rotten. So there was bacteria in there. And it was pretty shocking that anybody, anything, can survive those conditions. And so with a closer look, years of studying this bacteria, isolating it, taking it into the lab and starting to figure out, well, who is this guy that can survive sterilization with radiation? I mean, that's unheard of, right? And what we realize here is that, just to give you some perspective, our own cells, human cells, if we are exposed to 5 grays, this is actually 50 times the dose of an X-ray. So you're pretty safe when you're doing an X-ray. So 50 times that level is what our cells will start like. There are bacteria that we know it that would actually be able to survive a little bit more than that. And then this guy can actually do 3,000 times any amount of radiation that a human cell can survive. So if you radiate our cells with the levels that that guy can survive, we won't even get it close. Right? 3,000 times more than anything we can bear, and this guy is happy and can survive it. And so not only can it survive radiation, but actually extreme dryness, extreme stresses, UV light, you name it, and he seems to be able to do it. So one of the nicknames that is gotten in the bacterial world is Conan the Bacteria. And you can probably understand why. Right? There are other articles that will tell you that if you wanted to really send bacteria somewhere foreign on a ship, that's your guy. Right? So there's a lot about this guy that we don't understand but seems to be pretty special. Okay? Now, the remarkable thing about this, why this is so interesting and so special, is because these high levels of radiation actually cause quite a lot of damage. And that's why we're very careful when we use radiation in the hospital and in medical treatments, and we keep those doses very low. The reason why they can cause a lot of damage is because they induce these processes, it's a stress on our cells called oxidative stress. And what oxidative stress pretty much is literally upping up, right, increasing the levels of oxygen radicals that we have in our cells. So these are pretty nasty, very reactive oxygens that pretty much are going to go and scavenge and start changing every type of makeup that we have in our cell. And it turns out that these things happen to somewhat of a moderate level every day that we're alive. That's why with aging, they could be accumulating, and aging has, one of the definitions for aging is actually a buildup of these oxidative stresses. But the point is there's all this oxygen in the cell now that is pretty reactive. And what it does, one of its targets, one of the things it likes to do, is actually go and damage DNA. Well, you're probably thinking that's not a good thing, right? I mean, we talked about DNA encoding all this information that then gets activated so that we can actually have traits that we need to stay alive. Well, when under oxidative stresses condition that are induced by things like radiation, what we see is that this DNA, it's very damaged. And one of the things it does when it's damaged, it can break it can change. And when it happens, pretty much, if it breaks apart, you can imagine those are very unhealthy cells. So what we've said here is that most organisms cannot survive radiation. They cannot survive these high levels of radiation because radiation induces these very stressful chemical conditions that actually damage a lot of their makeup, including DNA. And if DNA is damaged, pretty much anything that it encodes for, we can no longer make. And again, might not be something too foreign. I think a lot of people might be aware of this because we try to desperately stop these stressful processes in our cells, and a lot of us actually turn into antioxidants to actually combat some of those natural stresses that oxygen can cause in our cells that, again, are very natural, and there will be some level of it, but very, very little. Radiation actually amplifies that quite a bit. Okay, so then how does this guy actually do it? What is so special about this bacteria? So when we started studying this, one of the things that we realized is that past scientists had actually wondered about the DNA. And the question that had been raised was, well, here's D-rat, he's carrying his DNA, and he can undergo all these radiation and be very happy afterwards. Well, what is so special about how its DNA is processed? Why doesn't its DNA break? Why isn't it damaged? So what I'm showing you here is stained DNA. And if you see these little sharp bands here, this is how your DNA, the bacteria DNA, D-rat's DNA will look like before radiation. You see these sharp bands here? That's a pretty healthy DNA. When you radiate cells, this is why this is so toxic, because your DNA all of a sudden loses those sharp bands and becomes broken, and broken DNA looks like this, very smeary. Right? So you see it's not a little sharp band anymore, it's all this mess of stuff here. This is what's bad, right? This is what's not going to allow anything useful to be processed and made or encoded by this DNA. But what you'll be surprised to know is that what this bacteria can do that is very unique is after just 30 minutes after you pretty much bump it with all this radiation, it can recover its DNA pretty quickly. So you see these sharp bands come back pretty quickly. So its DNA seems to recover very quickly after you irradiate it. So for the next 30 years in this field, people looking at this bacteria, there was this notion that the special powers of D-rat really relied on the ability to fix DNA. If it damages, it fixes it well, and that's why it can survive high levels of radiation. So one of the things that happened was the ability to actually obtain entire genomes, entire sequences. And you're probably very familiar with those movie images where you see this big screen and people start scrolling letters down and you see everybody's genome. It's actually true, right? So maybe if you're thinking of your next birthday present or what to get anybody for their birthday, you can actually save $1,000 and you can give them their genome. It'd be a pretty cool gift. I would love that. Right? But what you can do is, we have lots and lots more genes than D-rat has. So for pretty much $100, right, you can sequence every single DNA, get all the information that this bacteria would have. And that's what the research started going in that direction, and they were looking for something special and that was encoded in this information of the DNA that was leading to its special radiation powers. The results were pretty disappointing. So what was found in the field was that if you look at all the DNA information, here's D-rat, it was pretty disappointing to figure out that all the DNA information was pretty much identical to every other bacteria that will die under the same levels of radiation. So this did not explain the fact that they are unique traits here that we see. It can survive much higher levels, but not in this case. So something seemed to be really missing here. This whole notion that DNA is just special, there's extra information, the information is special, started pretty much going down the drain. So we kind of got it really interested in this research, and we had a new hypothesis that we wanted to really test, or we wanted to think about. We started thinking about, well, maybe it's not really the information per se. Maybe it's the way that this organism manages things under stress. Has the same information, but maybe it's just a better stress manager. Maybe it can actually decide how it's going to use that information in ways that are a lot more beneficial under stress, especially when it's under radiation stress. Okay? And so one of the things then that we did is now not only can we get a lot of this DNA information, but we can very specifically look at just the portion of DNA that has been actively processed, right, to be actually into active genes. So information can be in your cells. And if there's something I want you to remember, it's that this whole DNA thing is pretty dynamic. Information can be there, but it doesn't mean you're using it. It doesn't mean it's active. It can just be sitting there. And so we wanted to sort of differentiate between just DNA information being there, which is what the experiments before us were trying to do. And we were trying to look at, well, what is the information that's being actively used when radiation hits the organism? Maybe it changes, right? And so one of the things that we started doing was actually conducting studies at very low levels, no radiation, and putting this bacteria, putting DRAT to its maximum radiation that it can take. This is how much it can take before it starts dying on us. And so we wanted to look at, again, that portion of active genes that these bacteria was using under radiation. And you'll be very happy to know, we were happy to know, that just like we can sequence all the information in the DNA, we have technologies now that for very little amount of money, we can also, if I'm standing right here, I can check out and figure out what my entire body is activating or deactivating as I'm speaking to you. We can do the same thing with DRAT. We can put it under radiation, and we can put it under no irradiation, and we can look at all the DNA information that is actively being used. What is actively being turned on or turned off during radiation? And so we did this, and these green bands that I'm showing you here, if you see a bright green band, that green band means that not only is the DNA information there, but it's in its active form. It's being activated. And we assume that if it's being activated, it's making some kind of useful trait that the organism can use, that this bacteria can use to do something that it's helping with irradiation. Okay, so we can see here, this is a cartoon of some of the data that we've seen, that when you put DRAT on the radiation and you look at its active genes, you see a lot more, these patterns change. You see green here, but not green here. You see things that are active here, but they're not active here, right? This is all black, the active form is not there. So again, same DNA, but very different activation patterns on the radiation. And so what we started to think about was, well, maybe what's going on here is that DRAT senses radiation, and when it senses a radiation, it actually activates special information. And the special information gives it very special traits that make it more radiation resistant. It survives radiation better. And so you can easily imagine, right, that there could be a lot of useful traits that could be activated to protect it during radiation. You can imagine here shields, membranes, you can coat it. It can do a lot of things creative to protect itself. You can also imagine that maybe even additional DNA repair capabilities you can actually have. Your toolbox can get stronger to do all those repairing that you need when you're being attacked by radiation. That could be a useful trait. So they are useful traits that the bacteria can actually activate on the radiation. And so the one question then that was pending to us was, okay, let's take this hypothesis and run with it. Maybe there's something special about it during radiation, but how does it activate it only when it's under irradiation, right? Why aren't these traits active under any other conditions? Why can't we see it under any other conditions? So one of the last things I want to tell you that we've started to realize is that the conventional DNA theory, again, is that we just have lots of information, and that within information there was a belief for a long time that there was a lot of junk in our DNA, that it was just there to kind of be there, right? And one of the things that we now know is that this junk actually may be something useful. But back when we started doing a lot of this work, it's like, well, this junk is in our DNA, and it's junk because it doesn't encode for any traits that we can see. So it's no contributions to these traits. And so one of the things that we started to say was, you know what, let's in fact take a look at the garbage junk that people think, and let's see if there's anything meaningful there. And one of the things we see is that there's a pattern to that quote-unquote junk. And so here is radiation. These are, which I'm showing you images of what the junk looks like. So if you see a band, the junk is there. If there's no band, the junk is not there. And what you can see is that if I take my DNA and I just look at the areas that are active of my DNA, I find a lot of these junk that I can actually see, but I can only see it during radiation. So this junk, it's a pattern that you can only see on the radiation. So then we say, well, maybe is it really junk? Maybe there's something useful about this DNA that we are seeing that we thought was just kind of junk before. And to make the story kind of short here, what I'll tell you is that this junk turns out to be controllers. They are not encoding for traits, they're not encoding for thicker membranes, they're not encoding for all of this, but what they're doing is that they're activating specific regions of these genes here that provide useful traits for DRAD to survive radiation. Right? That's what they're actually doing. Now, when I think about this, right, in summary, what I've told you that DRAD seems to be doing, and we're still working on a lot of these theories in our lab, but what we think is going on here is that it has its normal DNA. And again, we haven't talked a lot about this kind of lighter box here. This is a so-called junk that people normally ignore. And so this junk within the DNA becomes visible or active only during radiation, and then it actually processes. A lot of helps with the processing of these genes that activate useful traits. Now, what I'm telling you is actually something that you've heard a lot about in the news. It turns out that these controllers or junk is what we call regulatory RNAs. And the only thing that I want you to realize about this is that these have been one of the most recent breakthroughs of biology. And the reason for that is that we actually, in this mechanism parallels what our human cells can do. And what is pretty powerful about these controllers that are only visible or are only present during the stresses is that it's just not enough anymore to know that we have DNA and that we're encoding information. That doesn't tell us the full picture by far, right? What we actually want to know is how are the traits that we could exhibit could be activated, right? What would activate? If we are prone to have a disease, what conditions would activate it? What stresses would be activated? What environmental stresses? What things that we might eat would activate these things? What things in our environment? What types of other types of social stresses? This is the whole way now that we're thinking about our genes. So it's not just enough to know what's your gene sequence. What we really care and want to know about is like DRAD. When are those genes activated? What are the types of signals, triggers, things that we can activate it? So there's this very active area of research on understanding gene activation. When do we exhibit those traits? It could be good traits that we can induce for ourselves. It could be bad traits, but when do they come out, right? And so we still look up to extremophiles like DRAD for a lot of answers to a lot of the challenges that we're facing. And I just want to tell you about a couple here to close. And so, again, as I alluded to earlier, one of the challenges that I think our generation and the next generation would really face is to solve this question of, is there life in other planets? How can any form of life be sustained in other planets? Well, maybe an organism like DRAD has some answers for us, right? We know that one of the reasons why we've always doubted life in planets like Mars or other planets is because they have a much thinner atmosphere than Earth. So we've always wondered, there's no way human life, there's no way any life as we know it can actually be sustained under those situations. Well, now we know one organism that can do it. So perhaps this opens doors to how we think about the universe. The other big question that we're going to have to solve in the coming up years, it's our continual degradation, continual solutions to human health. Right? Increasing stresses, things in our environment, things in our world, stresses because finals are coming, right? So what do these organisms know about handling stress and about surviving extreme situations that we can learn from? Maybe they have a lot of clues that our trillion cells can do to actually, can use to be more disease-free or avoid diseases. And so one of the things that we could learn is if we can actually borrow these shields, figure out what they're doing, or these helmets, right? Or these repair tools. Maybe we can mimic some of that in our own bodies and slow down diseases or be able to treat them. Lastly, right, radiation is one of the most common ways that we have to treat cancer. And there's a downside to that. if you have had any friends, any family, anybody going through the treatment, right, it's extremely difficult to see a patient go through it. It's devastating to our cells. It's a trade-off. The only way we know, or one of the few ways we know to kill cells effectively, is by throwing a whole bunch of radiation on them because we want to kill them. The problem is we're also killing a lot of things. So if we can learn from this organism a little bit about how to protect ourselves against radiation, maybe these kinds of therapies can be more effective. So there's a lot of ongoing research on this organism. And one of the things that you might want to know is that they are current mouse models, right? People are testing some of these features that we've discovered in DRAD to see if we can actually help mice models undergo chemotherapy in a more effective way. But in essence, what I want you to walk away with is a few things tonight. I want you to appreciate that microbes are really a huge group of people. I'm sorry, organisms. They're actually probably a lot more diverse than people. But they're a huge, I think of them as people, but they're a hugely diverse group of organisms. They are a hugely abundant way of life. And that the ones that we hear about that get all the bad press is just a very tiny fraction of the ones that are around us every day, that you're walking around with, that you're running around, that you're coexisting with every day. They are good microbes. There's a few bad players. Right? I also want you to realize the dependency that our society has really created on them. And I want you to realize that there's so many of them that we don't know that we can learn so much from to really make our lives a lot better. And then I want you to realize or walk away with this notion that one of the things that we are really being helped by microbes to do is to really re-examine the way ourselves, we process our genome, we process our information. It's not enough to know it, we can sequence it, but that's not going to be the end-all be-all. It's still going to be a lot about, well, when do we express what? What will put us ourselves at risk of diseases, of expressing bad traits? What can we do to ourselves to actually shield and protect and bring on good traits when we need it? So those are the main things I really want you to walk away today. And with that, I just want to close and tell you this is our research group. I think they did a nice work out there. These are some funding agencies that are funding some of this work. And you might notice, if you will see some of these acronyms, that the Defense Threat Reduction Agency, so the Department of Defense is very interested in DRAD. Because as you could imagine, they really want to be able to defend ourselves, defend our soldiers, defend anybody against really big environmental threats. So they really want to know these secrets. So they're one of our biggest funders for this research. And I just want to highlight in particular people that work in this area. These are grad students in the lab. So this is, Chen is one of my grad students who you might have met out there. He is really one of the guys that's working, has started a lot of this research in our group. And I just wanted to tell you that the reason why he got into microbes, or that he's a microbe fan, is because of the potential for engineering. So he comes to us from Taiwan. Another person is Justin. He's pursuing a master's, so he's another graduate student from biochemistry. And he got into this field, or he's a fan of microbes, because they make industrial and medical products. And Kevin, who I didn't talk a lot in detail about his work, he is one person that's working in the other side of the lab, which is trying to sort of understand a lot more of these oxidative stresses on human lung cells. He comes to us from Kentucky, and he's a microbe fan because he's interested in life and how life works, and that's what keeps his enthusiasm in this field going. We also have undergrads in our lab. So this Respina has worked with Chen for a long time, and she got interested in microbes because she heard from a very young age that she wanted vaccines were powerful, and she wanted to contribute to that. And the last person I want to feature is our high school student from Westwood High School, and this is a phenomenal student who has worked with us for summer. He's coming back this summer. And so he, at such a very young age, is fascinated by microbes, and he tells me it's because of all the applications he foresees. So Nick will be a student here after he graduates high school in the fall. Anyway, with that, I really want to thank you for your attention, and I can take a couple of questions, I guess.
Okay, for questions, we're going to ask you to line up by this microphone stand here. And if I can have your attention for just a second, if you could maintain quietness while we're transitioning to questions. I have an announcement. Better check, your school bus is waiting for you, so if you can quietly sneak out, we will begin questions. All right.
Thank you. I was wondering, with those superbugs you were talking about, could they withstand a nuclear explosion?
Oh, that's a great question. I love it. The answer is yes. DRAD is thought to be one of the few organisms that will stand nuclear explosions. And you know what's really cool about it? That it's used in sites, places where there's been nuclear explosions. You can take it there and it helps to clean the site. So it swallows all those metals and all that nasty stuff there because it loves it. It loves to grow on natural explosion sites.
Oh, cool.
Yes.
So the virus, well, I have two things. So first of all, the virus is 2/100 of, I forgot the word.
Microns?
Microns. And the bacteria is 1 micron. How many microns is Outbreak?
How many microns is what?
Outbreak, the movie.
Oh, I see. Okay, that's a great question. Okay, so let's get something a little bit clear here. Microns is a measurement of length. So if I put my hands out here versus here versus here, this means little microns. This means big microns, lots of microns. So that's a unit of length. It's a meter, right? Now, the movie, it's time, right? So you can say that's 2 hours, but you can't measure that with a metric tape, right?
Right.
So we can't measure a movie with a metric tape, but we can measure these things with a metric tape. So it's hard to compare them.
Mm-hmm. Okay, cool. And my second thing, so as you can see, I'm losing my voice. Is that bacteria or a virus?
Well, there could be a lot of things. It could be you just had to have a very exciting day today, and your vocal cords are just sore. But it could be a bacteria or it could be a virus. But when you lose your voice, those are just signs that you might be sick, but you may not. Tomorrow you'll be fine. But it could be both. If you have a cold, that's like a virus. If you have like an infection where a bacteria, you ate something and it was kind of bad, that could be a bacteria.
Yeah, I do have a cold. So.
Okay.
How do you make better bacteria in your body?
Oh, that's a great question. Well, you are asking a question that people are spending lots of money to know. We don't know, but it's a great question. So our bodies, bacteria are naturally there. So what we can do to make them better is just eat things that will make them happier, that won't kill them, and sort of maintain them alive in our bodies by what we eat, by exercising, by not stressing too much and producing things that will kill them. But they're naturally there. They're naturally there from the time you were a baby. And they're still with you. And you want them with you.
Okay.
Are there more good germs or bad germs?
Absolutely. There's only a very, very small portion of germs that we know that are bad germs. Much, much of the germs we know are good germs.
Okay.
All right, I'm going to have to adjust this or else I'm going to have to get down. But it's okay, I can get down. All right, so Dr. Contreras, this has been a fascinating talk. I was kind of intrigued when you said that we could learn from the repair and protection mechanisms that DRAD has and apply it to human cells. Now, I'm not a microbiologist, I'm a plant biologist, but I know that bacteria DNA and genomes are very different, like they're circular, they're much simpler. Is it at all feasible that we could apply the same genetic repair mechanisms it has to eukaryote cells?
Well,
I mean, like, how feasible would that really be?
Yeah, that's a good question. And so there's two ways to answer that question. One thing that you could do, you're basing it on the fact that we would have to go back and look at the genomes and make genome comparisons, and you pretty much get to a doom point if your eukaryotic cells don't share the same DNA or the same factors that these bacteria do. But what's actually known is that they are metals, they are things that are pretty much in any cell. So it could be a simple thing as we realize, which we're, there is some research on this, that the charge, it matters. So things like, magnesium or manganese, things that are positively charged would actually protect your molecules. So you can actually take those as supplements. And that's what they're doing with a lot of these mice actually. So they're giving them a lot of these cofactors just because we realize that even having positive charge to balance the chemistry of your DNA, and the chemistry is very similar in all organisms, that could help. So there could be really simple things that we can extract or, without having to necessarily mimic a mechanism.
All right. Thank you. That was very enlightening.
Thanks.
So I have 3 questions.
Okay.
What was the disease in the outbreak? Was it actually a real disease?
Well, it was a virus. They usually make up these things. So it was a made-up virus, but it was supposed to be a virus that's transmitted from monkeys. They like monkeys in these movies.
And what would happen if you ate a D-rad?
That's a good question. I don't know that I would try it, but it's not known to cause disease. So I don't think much would happen.
DNA?
Huh?
Could it give you its DNA?
Even if it does, well, chances are it won't because it's very shielded and protected, so it just doesn't release it. But even if it did, I don't think it can really harm you. It doesn't really do anything. It may not taste good, but it doesn't really do anything.
And what would happen if you put a D-rad in a black hole?
Well, that's an experiment that I don't think has been done. It would be an interesting one, but one of the things that D-rad can do is, remember those big shielding things that it has, those thick membrane coats that it has? It can actually protect itself against a lot of pressure, right? And so if you change, putting it in a black hole, the most impact you can probably have is things like maybe changing a little bit the temperature that it's used to or the pressure that it feels. I think you'll probably do very well.
But black holes pull stuff apart.
Well, we would have to test it out.
Right,
but I would put some money on him.
Thank you.
So you seem to have said that D-rad is not dangerous to humans. Do we have any way to kill him not that we'd want to, but if he got in our bodies without killing ourselves?
If you wanted to kill him inside your body, it will be a little bit harder. Because any way that we have to kill bacteria, you would have to develop a very special antibiotic, a chemical, that would bind it and kill it. But anything physical, like hitting it or bumping it or putting radiation or putting higher temperatures, probably will kill you first before it kills D-rad. So you probably don't want to do that. So I would go the chemical route. Building a chemical especially hits it, but not you. And that's hard.
Okay, and also, have we discovered any bacteria that can survive in a vacuum?
I don't know that we have. But what's interesting about your question is that one of the ways that we are testing how to take flights out of Earth, as in space, is by putting bacteria on chips and figuring out, taking information through robots and figuring out if life forms can actually withstand it before we send any humans out there. So bacteria are being sent out, and they're also being sent out so that we understand potentially the effect that these things can have on astronauts as they travel across space. So we are doing those experiments, but I don't know that we've done, not as in me, my lab, but other people are doing those experiments. NASA is very interested in that.
Part of my question was, would they be able to survive without air or any gases around them?
Well, air, it's bacteria, a lot of bacteria actually hate air because they don't like oxygen. So they actually grow better without oxygen. And we work with one of those in the lab that actually makes ethanol, like a biofuel type of thing. And it actually hates to make ethanol with oxygen. So we keep air away from it so that it's a better producer of what we want to use it for.
But would it be able to survive without any gases at all?
Well, it needs nutrients. It needs nutrients, right? But they need to eat something. But what they can eat is a very large repertoire of things. They can eat a lot of things, but it doesn't need anything. Air, not so much. They don't really need air.
So if you put it in space with a slab of metal, it might survive until the metal is
Absolutely
gone?
can survive. In fact, a lot of them love metal. They actually like to live on metal because metal is like part of their diet. Like it's what they like. It's like pizza. So they like metal, yeah.
Okay then.
Did you say earlier? You said something about like radiation being the thing that caused aging and stuff?
Yeah. So yeah, I can see that.
Is there a way that if you like mutated T-Rad's genes to get into your body instead, that you would be able to stop aging?
I love it. There could be. I love it. It could be.
And also, do you know if, like, D-Rad could survive in something kind of like, let's say, like Pluto or something?
As I said in the talk, if you look it up, so when you get home, Google it. And Google bacteria that can survive away from Earth. People are betting that he can. Okay. But it hasn't been done because Pluto's kind of far to take it there. I think that's more of the inconvenience.
Yeah, true.
Yeah.
So I think the end of the line now will be the last question, so no one else needs to get up online.
Okay, two questions. One is kind of piggybacking on the question that another student asked. So you mentioned that they could thrive in nuclear disaster areas. So do you know of any studies that they've done on like Chernobyl? Because I recently saw an article that mentioned that they saw that there is no decomposition occurring because of the radiation that has killed all the primary decomposers. So do you know of, or would you think that this bacterium would be able to kickstart that decomposition cycle?
Interesting. You know, I don't know of that particular site, but I do know that it's being used heavily on sort of cleaning contaminated sites. Both contaminated with toxic metals and contaminated from residues of radiation, explosions, or things going wrong with radiation. But I would have to look that up.
Right.
I don't know in that particular site it has been tried.
Okay, cool.
But thanks for bringing that up.
Yeah, I just thought it'd be good. And then the other one is I'm curious about, because my actual major was industrial microbiology. So I remember doing a lot of studies where we used BLAST to compare different genomes, and when I saw that different arrays of DNA activation in DRAD when it is radiated, have those genes been compared or like blasted against the human genome to see how many of those actually are the same or similar?
Yeah, that's a good question. So I almost want to turn you over to Chen, who's become, you know, an expert on this stuff. But he's looked a lot about this. And what it seems to be is that they are genes that are different, right? Because obviously, you know, DRAD has a much smaller genome than our genome. So we have 20,000 genes. They have 3,000 to 4,000 genes, right? So it's smaller. But what is thought right now is that there's nothing special in the actual information. It's more actually the one theory I presented today. It's sort of the how you use that information and how you sense your environment and coordinate those needs to actually express things that are going to be helpful to you. So it's more on this sort of gene regulation and control than that. The other theory that I didn't talk about today, it's more the protection of the proteins. So once those genes become active, right, those genes are going to be traits, and those traits are often proteins. And proteins are actually shielded by compounds in DRAD that humans do not seem to be using those similar mechanisms. And that's what they're trying to borrow to protect human cells.
So like you would be saying that we do have the capability of producing those proteins, but we just don't have the, I guess, the guide for them to be activated in that sequence to protect us?
That's what I personally think.
Okay.
And I think that part of what's going on here that I didn't really emphasize a lot is that this guy is very good at sensing the environment.
Okay.
Is very in tune with this radiation thing, and we might just not have those sensing capabilities that would connect it to an output that would be really beneficial for us. If we can connect those things, if we can sense it and then activate useful things, I think we could be in a much better place. But we've never had the need to develop those mechanisms, so we never did.
Cool. Interesting. Thanks.
So it's great that you all have so many questions apiece, but I think for us to give everyone a chance, please pick your best question to ask, Professor. Pick your best one.
Okay. Do you think that it's at all likely for there to be life on Titan? Titan. And what do you think its special traits it would need? What special traits would it need to live on Titan if there was life on Titan?
I'm sorry, to live where?
Titan, one of...
You guys
I believe it's...
You guys have to tell me what Titan is first.
Titan is a moon. I forgot whether it was around Jupiter or Saturn.
Moon. Yes, the moon.
Yeah.
Well, that's an interesting question. You know, I think that to a certain degree, a lot of these organisms or bacteria, they need a cycle. They need to kind of a routine to wake up, you know, and do things and receive cues from their environment. So I'm not sure because I don't know that much about the moon. I don't know what kinds of things they can find in their surroundings to actually, right, you call life, I mean, in a very basic way, when you're interacting with something and responding, right? If I'm pinching, I'm gonna move my head. That's my hand, because I feel it. So I don't know what kind of cues there'll be there in the moon that any organism can feel and work off. But I think if we knew a little bit more, it would be easier to imagine.
But do you think that there could very well be life on Titan?
Yeah, there could be. There could be life, and there could be organisms that we've no idea, we've never seen. We can't even imagine, right? Until 60 years ago, we didn't know that anything could be radiation, you know, could live through radiation. This is just 60 years ago.
Even though Titan has an extremely thick atmosphere, radiation I don't think would affect it all that much.
That's right. That's right. We would have to know a little bit more about Titan, and then maybe organisms that could survive that environment.
Okay, thank you.
Thank you.
You talked about how many microbes have a mutualistic relationship with our own cells, as well as a lot of other biological cells. Are there any instances where microbes have mutualistic relationships with other microbes?
A lot. A lot. In fact, one of the things that the Army wants to understand a lot about is when soldiers get injured. Usually the worst injuries that never close, right, that really kind of lead people to bleed all over, are because there's not one single bacteria there. They are 2 or 3 together. So there's something they do together that one cannot do on their own. And we're getting now towards really figuring out what these communities are doing. And it's the same thing in our bodies. There are these communities where they don't work on isolation. They kind of work together like a little team or community, right? And they help one another. So if I make something that is garbage to me because I don't like what I'm making, you might be able to eat it and it may be really good for you. And then in turn, you can make something that can help me grow better. So there's this sort of what you're calling the symbiotic relationships that are all over the bacterial world. And I think we're just scratching the surfaces of those. But that gangrene and the bleeding and the intense bleeding, it's one example that's it's of a lot of concern to the Army, treating those.
Thank you.
Can there be other microorganisms on microbes?
I'm sorry, can you say that again?
Could there be other microorganisms on microbes?
Ooh, that's a good one. Can there be any other microorganisms of microbes? I don't know of any, but there could be. I don't know of any. I mean, I know they work a lot together, but they could be. I know there are microbes in plants. I know that there are viruses in insects. I know, but I don't know. I can't think of a good example of that. but they could be. They seem to be pretty independent on their own. But I'll have to think about that. That's a good question. What do you think they would do if they were there?
They could help the microbes.
They could help the microbes, yeah.
Survive.
That's right. I'll have to think about that.
I think I can just reach it this way. So the DRAD was found originally in radiated meat. Does that imply that radiation in meat selected for DRAD? And if so, does that mean that radiation in meat is not such a great idea?
Well, I think radiation, somehow that meat got contaminated. And I think it's very unlikely that radiation of meat won't kill bacteria. Now, what probably happened after that episode is that doses went far off even to kill something like DRAD there. Or do you have to do a test for that kind of bacteria in there? But I don't think that really has as many major implications for the meat sterilization process as it does for really us being able to understand what aspects of life could sort of survive these conditions. But I think the meat industry probably, that was a wake-up call on that. And they have, now we know, right, that this may require a few more cycles. But so often, right, we hear people say they're concerned about food safety. We hear things in spinach. So I mean, things could definitely bypass, but the most probable cause for a lot of these processes to sterilize our food is mutators. It's bacteria that would go there and just kind of spontaneously come up with something even cooler in their DNA that encodes for something that we never predicted they would. So anything we have to deal with them is all of a sudden goes down the drain because they develop new arts and new forms of doing things they want to do.
So you're killing the cells with radiation that don't have that mutation, but you're selecting for those.
Absolutely.
Okay.
Absolutely. Exactly. You would be totally selecting for cells which is what the antibiotic problem is so interesting. Because you throw an antibiotic there, does that mean you're really killing every bacteria, or does that mean you're actually selecting for the ones that don't get hit by that antibiotic? And we often use combinations for that reason, and that field, it's in a lot of trouble most of the time. Yeah, that's a really good point.
Why did they want to take a monkey?
Why did they want to take a monkey?
Oh, out of its habitat.
I know. Yeah, it's a hard thing to do. I guess the reason why people like to take monkeys out of their habitat, they don't like it, but they feel it's necessary is to help humans, because monkeys are believed to be really close to humans. So we start testing mice and we start testing bacteria, but when we're starting to figure out that something might help humans that are sick, we have to go and test it in something that looks more like a human. So they'll take a monkey.
Okay.
She remembered.
I thought you were just trying to bring the average number of questions per person down.
How do they move around place to place?
Well, remember they had that little tail? I don't know that I can. Well, maybe I can find it in
I remember.
You remember? It had a little tail. They move that little tail around, and that's how they kind of move from place to place.
Okay. Do they poop?
Well, yes and no, right? So what we call poop is a very human form of getting rid of nasty stuff. They have their own ways of getting rid of nasty stuff that they make and don't need. So yes and no. But it's not the poop you probably are thinking of. That's a good question. That's a very good question.
Excuse me. How many microbes can surround one cell?
How many microbes can?
Yeah. Surround one cell.
Can survive one cell? Surround.
Yes.
Ah, that's a good question. Well, if you take one human cell One. And in your body, you have lots and lots of lots. You have trillions of them. That's a big number. If you take one of those guys, you can probably fit about 10 to 20 bacteria there.
Cool. Thank you.
Do you think there is like a DRAD DNA gene in the human body that we just don't know how to activate? And if there is there a way we could use technology to activate that so we could have the same powers as DRAD?
Yep, that's what we're doing in our research. There could very well be either one gene or one system or one type of systems, and maybe there's one that you can combine with another one and do what DRAD does. And that's exactly what we're working on in our lab. So we should hire you.
Thanks, I'll think about that.
Okay.
Thank you, guys. We will see you in the fall. If you're on our email list, you'll soon be getting word of who we're having visiting us this coming fall. Have a good night.